Okay, I'll be very quickly try to explain why branched peptide, but the story is really too long to be explained into detail. So now, many years ago, we discovered that when peptides are synthesized in a branched form, which can be easily obtained by chemical synthesis, using a core made by lysine amino acid, which is schematically reported here. When these peptides are synthesized in this form, they become extremely resistant to proteases, and particularly to peptidases, which means that they acquire features that can be useful for in vivo use. So you see here that when we discovered that, we use this technique and we move to monomeric to branched peptide, and for several applications which you can see here. Oh, sorry. Okay, so in this presentation I will show something which is regarding the application of branched peptide in cancer, and in a very new project which we started thanks to the Tuscany Ecosystem regarding possible bone regeneration. Then in the next presentation you will hear about another branched peptide which was used for years for treating bacterial infection. So regarding the target, the cancer targeting by branched peptide, many years ago, we synthesized a sequence which was practically derived from a natural peptide, but the story is really too long. And because we realized that when this peptide was synthesized in a branched form, note, only when it was synthesized in a branched form, not in a monomeric form, it was extremely specific for recognizing many different cancer tissue, as you can see here, and in many different cancers. This was published now many years ago: colon, pancreas, urinary bladder. Using the peptide for staining the tissue, this is what was obtained. So the cancer tissue was the staining of cancer tissue was extremely higher compared to the staining of the healthy tissue from the same patient. We collected many different tissues, and so we could analyze we could perform a statistical analysis on what we could see in the microscope. And later on, we discovered which the target was, and that was interesting because the target of this branched sequence were the glycosaminoglycan sulfated chain of heparan sulfate proteoglycans. And also thanks to our studies, it was demonstrated that many cancers, they express extremely highly sulfated heparan sulfate proteoglycan, much more than healthy tissue. So they overexpress these, but more than that, they oversulfate these heparan sulfate proteoglycan. So we used that, thanks to this cancer selectivity, we thought that it was possible to use that peptide for targeting cancer, and we, of course, we started with analyzing what we got in the cell for cytotoxicity, but then quickly we moved in vivo model, and this is a model where we use this branched peptide conjugated to paclitaxel. And we compare what we got with the name of this peptide is NT4, conjugated with paclitaxel, and this is what we got when we compared not only with the control group, but even with group treated with the same concentration, the same amount of paclitaxel. So the use of this peptide conjugated to paclitaxel in this orthotopic breast cancer model, mice model, was really very good, and in some of these mice we have clearance from the disease. But then we knowing that the target was the heparan sulfate proteoglycan, we also wanted to use those peptides as a tool for analyzing the function of these molecules in cancer, which is not completely known. And so, since we had a tool which was specific for the heparan sulfated chain of these membrane proteoglycans, and those are known to have a role, not clear how much, not here how autonomous, particularly in cancer cell adhesion and migration. And so we use the peptide, the branch peptide, now unconjugated, so with nothing conjugated to, for analyzing the effect on cancer cell migration. This is what we got with different cancer cells. This is the punctus adenocarcinoma, and these are the very famous breast cancer, human breast cancer aggressive cell line. And by adding the peptide, it was clear that the migration was lowered. And then we also analyzed how this peptide was effective in another in vitro model for cancer invasiveness, which is the ability of the cell to make colonies in vivo. So you just seed the cell at very, very high dilution. You wait for some days, and then you measure how many colonies are formed. And in the presence of the peptide, colonies in both the punctus adenocarcinoma, and particularly in the breast and also in the breast cancer cell line, the number of colonies was much, much lower. And so we started to try to understand what these heparan sulfate proteoglycan were doing, and which was, and we are still analyzing, which the role is in cancer cell migration. And what we noticed is that by using the peptide, but also using an antibody which is commercially available, which recognizes heparan sulfate on this heparan sulfate proteoglycan, when you have migrating cell, those sulfated chain stay on the border. So they are much more expressed on the border of migrating cell and much less in the interior. And we also wanted to see which was the proteoglycan which was carrying those heparan sulfate chain, and those are different in different cell lines. And in this case, we had the Syndecan-4. I don't have time to explain what Syndecan-4 means, but Syndecan-4 is one of these heparan sulfate proteoglycan which can be directly involved in a signaling, because it has several intercellular domains which are known to be linked to different signaling. Not exactly how, but they are known to be related to the intracellular signaling for even, and particularly for the signaling for the organization of the actin cytoskeleton, which means migration. And okay, so this, I will very, very quickly move to the other, to a completely different topic, which is the new project that I told you we initiated thanks to this Tuscany Health Ecosystem program, so it is pretty new and completely another thing. So we wanted to see whether it was possible to select branched peptide, which can then be synthesized in a branched form, which we usually do in our laboratory. And the target that we wanted the target of our choice was sclerostin. I don't have time to go into detail of the signaling, but sclerostin is an inhibitor of an important signaling, which is Wnt. And by inhibiting Wnt, sclerostin is one of the most important regulators of bone regeneration. So as long as it is there, the bone regeneration is off. And in order to increase the bone regeneration, you have to get rid of sclerostin. And so what we wanted to do was to select a peptide able to bind to sclerostin and eventually interfere with the role of sclerostin in the Wnt signaling regulation. So inhibiting sclerostin means to switch off the Wnt bone regenerating signaling. And for this reason, sclerostin is already considered as a very important target for possible therapy for osteoporosis. So I will very, very quickly say, because we are just at the beginning of this study, so what we did was what we did several times for our targeted peptides. So it was to select the peptide by phage display. We spent some time for setting up the methods which could enable then the possible analysis of the function of peptide that hopefully we could select. But since the beginning, we also performed two different panning, so the two different selection of these phages. One was the classical selection by elution buffer by pH, and the other one was was a competitor panning because sclerostin is an heparin binding protein, and it binds to LRP receptor, and as many binders of the LRP receptor, they also bind to heparin. So they are heparin binding protein. So we also performed a selection by competition. So the phages bound to the antigen were detached by adding heparin. So the heparin could bind to the immobilized sclerostin, which was used for the panning, and hopefully we could select those phages that are that bind heparin, which means that hopefully they can more probably bind also LRP receptor. And I would like to tell the end of the story, but unfortunately I cannot because we are still working on that. We already have some peptides. We synthesized in a branched form, and we are just in these days currently analyzing the function of this peptide by analyzing the effect on Wnt and signaling, and before that the binding and the affinity of this peptide. I think this was my last slide, so I don't have the photo of the collaborators, but many of them are here, so you can see them. And so I want to thank all the group and, of course, you for your attention. Thank you.